In March 2019, a Mossad case officer named Yael stood in a sterile laboratory outside Tel Aviv, holding an evidence bag containing a cloth napkin. The napkin had been retrieved from a restaurant in Istanbul 72 hours earlier. It had been used by a man who entered Turkey on a diplomatic passport, stayed for 11 hours, met with 2 individuals in a private dining room, and left on a flight to Damascus before Israeli surveillance could confirm his identity. The napkin had been handled by a waiter, transported in a backpack across 2 borders, stored in a refrigerated case, and delivered to this lab under chain of custody protocols that didn't officially exist. Yael wasn't certain the sample was usable. She wasn't certain it belonged to the man her field team believed it belonged to. And she wasn't certain the entire premise of the operation, that human genetics could be weaponized as an intelligence tool, would work outside a controlled environment. What she did know was this: if the napkin contained viable DNA, and if that DNA matched the biological signature of Abbas Nilforoushan, a senior commander in Iran's Quds force, then Mossad would have confirmed his presence in Istanbul without ever seeing his face. And if the method worked once, it could work again. Not just for one target, for an entire network. This is the story of an operation that began with a hypothesis about what the human body leaves behind. It's a story about decisions made under incomplete information. And it's a story about what happens when an intelligence agency discovers that the evidence it's been collecting may not mean what it thinks it means. The operation didn't begin with genetics, it began with failure. For 3 years, Mossad had attempted to map the individuals who surrounded Iran's supreme leader, Ali Khamenei. The objective was straightforward: identify who had access to him, track their movements, and understand their relationships. The challenge was structural. Khamenei's inner circle operated within security protocols that made traditional intelligence collection nearly impossible. Members changed phones every few days. They traveled without itineraries. They communicated through couriers whose identities were unknown to Western intelligence services. By mid-2018, Mossad's intelligence directorate had compiled a list of 32 individuals believed to have regular access to Khamenei. The list was based on photographic analysis, defector testimony, and signals intelligence, but believed was the operational word. Mossad couldn't confirm which of those 32 were actually present during decision-making meetings. It couldn't map familiar relationships. And it couldn't verify whether certain figures photographed at public events were the actual officials or body doubles. Yael, a mid-level case officer with a background in molecular biology, proposed a different approach in a planning session in November 2018. The idea was simple: collect biological material from suspected members of Khamenei's circle wherever they traveled outside Iran. Extract DNA from that material. Use genetic sequencing to establish biological identities that couldn't be obscured by encryption, disguises, or operational security. Build a database that would allow Mossad to track individuals not by intercepting their communications, but by confirming their physical presence through cellular evidence they didn't know they were leaving. The proposal was approved, but not enthusiastically. Leadership understood the concept. What they questioned was the feasibility. Genetic material degrades within hours in uncontrolled environments. Chain of custody failures could render samples legally and operationally worthless, and there was no existing infrastructure within Mossad for handling biological intelligence at scale. The agency had labs for analyzing intercepted documents and seized electronics. It didn't have labs designed to process saliva from a coffee cup retrieved by a cleaner in a Beirut hotel. Yael was authorized to run a pilot program targeting 3 individuals: Abbas Nilforoushan, Hossein Taib, then head of the IRGC's intelligence organization, and Mojtaba Khamenei, the supreme leader's second son, and a figure believed to be involved in operational decision-making. The collection phase began in January 2019. Field teams were deployed to 6 cities where Iranian officials were known to travel: Istanbul, Beirut, Damascus, Muscat, Baghdad, and Caracas. The teams included Mossad operatives embedded as hotel cleaning staff, restaurant workers, airport maintenance personnel, and private drivers. Their instructions were specific: retrieve any item that might contain biological material, drinking glasses, eating utensils, towels, discarded razors, cigarette butts, or toothbrushes, without alerting local security services or Iranian counterintelligence. The operatives were not told why they were collecting these items. They were told only that contamination was unacceptable, and that each sample required documentation, location, time of collection, description of the individual who used the item, and chain of custody from retrieval to laboratory delivery. Within 8 weeks, Yael's team had collected 47 samples. 16 came from hotel rooms, 12 came from restaurants, 8 came from vehicles. The remainder came from airports, embassies, and a private clinic in Damascus. Only 11 samples were usable. The rest had been contaminated during handling, degraded by heat or moisture, or contained insufficient cellular material for sequencing. In 4 cases, the samples had been collected from shared items, plates handled by multiple people, glasses washed and reused, making it impossible to isolate a single genetic profile. Of the 11 usable samples, 3 were confirmed through cross-referencing to have come from the intended targets. 1 was the napkin from Istanbul. Another was a water bottle retrieved from a safe house in Damascus. The third was a disposable razor taken from a hotel bathroom in Mossad. Yael believed the operation was progressing as expected. Early losses were acceptable. The method would improve. The lab protocols would tighten. The field teams would learn what worked. What she didn't yet understand was that one of the 3 confirmed samples didn't belong to the person Mossad thought it belonged to, and that misidentification would force a rewrite of everything Israeli intelligence believed about who made decisions inside Iran's most protected circle. But first, the samples had to be sequenced, and sequencing required a lab that didn't exist. The samples were transported to a facility that had been constructed in 6 weeks on the grounds of a decommissioned military research station outside Tel Aviv. The facility wasn't designed for genetics, it was designed for deniability. If questioned, the Israeli government could claim it was a forensic lab used for criminal investigations. Officially, it wasn't a Mossad installation. Unofficially, every person working inside reported to the Intelligence Directorate. The lead scientist was Dr. Ron Shamir, a geneticist who had spent 12 years analyzing degraded DNA for war crimes tribunals and criminal prosecutions. Shamir's task was to extract usable genetic profiles from samples that were often incomplete, contaminated, or damaged by environmental exposure. A full human genome contains 3 billion base pairs. The samples Mossad collected rarely provided more than a few thousand usable sequences. Shamir had to determine which fragments were sufficient to establish a unique biological signature that could be cross-referenced against future samples or existing intelligence. The first sample processed was the napkin from Istanbul. Shamir's team extracted cellular material from the saliva residue, amplified the DNA using polymerase chain reaction, and sequenced the fragments that remained intact. The profile was partial but usable. It showed markers consistent with Middle Eastern ancestry and male sex. But there was no reference sample to compare it against. Mossad didn't have Abbas Nilfarushan's DNA on file. What it did have was a genetic profile from a second cousin of Nilfarushan, who had defected to Germany in 2014 and had voluntarily provided biological samples during debriefing. Shamir ran a familial analysis. The Istanbul sample showed genetic overlap consistent with a second or third degree relative. It wasn't definitive proof, but it was enough to assign the sample a provisional identity, subject Aleph 01, probable match for Abbas Nilfarushan. Yael requested confirmation through independent means. Mossad's Signals Intelligence Unit reviewed metadata from phones known to be in Istanbul during the collection window. One phone, linked to an Iranian diplomatic credential, had pinged cell towers near the restaurant where the napkin was retrieved. The phone's travel history matched Nilfarushan's known movements over the previous 6 months. The genetic data and the signal data pointed to the same conclusion. Yael entered the profile into the database. The first collection was a success. The second sample, the water bottle from Damascus, was processed in early April. Shamir's team extracted DNA from saliva residue on the bottle's rim. The sequencing produced a full profile, cleaner than the Istanbul sample. Shamir ran it through the database. No match. That was expected. Mossad didn't have genetic profiles for most of its targets. The Damascus sample was entered as subject Aleph 02, pending identification through field intelligence. Then Shamir ran a familial analysis. The software flagged a match. The Damascus DNA showed a sibling-level relationship to a profile in Mossad's holdings, genetic material obtained from Mojtaba Khamenei's younger sister during a medical procedure in Austria in 2016. The Damascus sample belonged to someone in the Khamenei family. but it wasn't Mojtaba. Shamir ran the analysis again. The results didn't change. The DNA indicated a male sibling of Mojtaba Khamenei. That left two possibilities: Mossad Khamenei, Mojtaba's younger brother, or Mostafa, the eldest son, who had died in 2011 according to Iranian state media. Yael pulled the collection file. The water bottle had been retrieved from a room in a Damascus hotel used by Iranian officials. The field operative had been instructed to target a man matching Mojtaba's description: mid-40s, short beard, minimal security detail. The man had been photographed entering the hotel. The images were processed through facial recognition software, which returned a 76% confidence match for Mojtaba Khamenei. But the DNA said otherwise. Yael reviewed the photographs again. The man's face was partially obscured by a scarf. The angle was poor. The lighting was inconsistent. A 76% match meant the software was uncertain. And uncertainty, Yael now realized, had been treated as confirmation. She brought the discrepancy to a meeting with the Intelligence Directorate in mid-April. The tone was clinical. If the Damascus sample didn't belong to Mojtaba, it likely belonged to Mossad. If it belonged to Mossad, then Israeli intelligence had misidentified which of Khamenei's sons was operationally active. And if Mossad was traveling to Damascus for meetings with senior IRGC commanders, then his role was far more significant than Mossad had assessed. One division head asked the question Yael had been avoiding. Was it possible the sample had been planted? Shamir answered. Planting DNA required access to biological material from a specific individual, the technical capability to preserve and transport that material without degradation, and knowledge that Mossad was conducting genetic collection in that specific location. It was possible, but it required a level of operational sophistication that suggested Iranian counterintelligence already knew about the program. Yael didn't believe the sample was planted, but she couldn't rule it out. And the doubt introduced a deeper problem. If genetic evidence could be planted, then every sample in the database was potentially suspect. The operation had been designed to bypass the weaknesses of human intelligence, sources that lied, signals that were encrypted, and photographs that could be doctored. DNA was supposed to be objective. But if it could be manipulated, it became just another vector for deception. The meeting ended without resolution. Yael was instructed to continue the program, but to verify the Damascus sample through an independent collection. If a second sample from the same individual could be obtained, and if it matched the first, then the identification would be confirmed. If it didn't match, the program had a contamination problem. Yael authorized a follow-up operation in Beirut, where Iranian officials were scheduled for meetings with Hezbollah commanders in early May. A field team was tasked with collecting samples from anyone traveling in a convoy associated with Mojtaba Khamenei. The objective was to obtain Mojtaba's DNA, which would clarify whether the Damascus sample had been misattributed. The Beirut operation succeeded in collecting two samples, a coffee cup and a cigarette butt. Both were processed in late May. Neither matched Mojtaba Khamenei. One matched the Damascus water bottle. The confirmation arrived in a two-page memo from Shamir's lab on May 29th. The Damascus sample and the Beirut sample came from the same individual. That individual was genetically consistent with being Mossad Khamenei, not Mojtaba. Facial recognition had failed. Field identification had failed. And for more than 2 years, Mossad had been tracking the wrong brother. Yael read the memo twice. Then she pulled the operational files on every intelligence assessment that had referenced Mojtaba Khamenei since 2017. There were 43 reports. 16 described meetings Mojtaba had allegedly attended. 9 detailed travel patterns attributed to him. 6 assessed his influence within the IRGC command structure. If even a fraction of those reports were based on misidentified individuals, then Mossad's understanding of Iran's leadership wasn't incomplete. It was structurally wrong. The realization triggered an internal review. A task force was assembled to re-examine every photographic identification of Mojtaba Khamenei over the past 5 years. The review found that in 11 instances, the confidence level of facial recognition software had been below 80%, but the identifications had been accepted anyway because they aligned with operational assumptions about where Mojtaba was expected to be. The genetic data had exposed a methodological failure. Mossad had been confirming what it expected to see, not what was actually there. But the deeper problem was strategic. If Mossad Khamenei was traveling to Damascus and Beirut for meetings with IRGC and Hezbollah commanders, and if Israeli intelligence had been unaware of his operational role, then there was an entire decision-making structure inside Iran that Mossad had never mapped. Mossad wasn't a peripheral figure, he was a blind spot. Yael was called into a closed-door meeting in early June. The question put to her was direct: should the genetic program continue, or should it be shut down before it produced more intelligence that contradicted what Mossad thought it knew? She didn't have an answer. What she had was a database with 19 genetic profiles, 3 confirmed misidentifications, and no way to determine how many other errors were embedded in years of intelligence reporting. The program was fragile, and fragility in intelligence work was the same as failure. The program didn't stop. It expanded. In mid-June, Yael received authorization to shift the collection mandate. The objective was no longer to confirm the identities of individuals Mossad already believed it knew. The objective was to map a network Mossad had never seen, using genetics to reveal familial relationships, travel overlaps, and operational clusters that traditional intelligence methods had missed. The decision was made under pressure. The Mossad revelation had created institutional doubt. If one of Khamenei's sons had been operating undetected for years, how many other figures were moving through the same blind spots? Leadership wanted answers. Yael was told to produce them. She designed a new collection protocol. Instead of targeting specific individuals, field teams would collect samples from any location where Iranian officials congregated: hotels hosting diplomatic delegations, restaurants near embassies, airports with direct flights to Tehran, and safe houses used by the IRGC. The samples would be sequenced and entered into the database. Familial analysis software would identify genetic clusters, groups of individuals related by blood or marriage. Those clusters would then be cross-referenced against signals intelligence, travel records, and photographic surveillance to build a biological map of Khamenei's inner circle. The first phase of expanded collection began in July. Field teams operated in 8 cities. Over 6 weeks, they retrieved 73 samples. 41 were usable. 16 matched individuals already in the database. 12 were new profiles with no prior intelligence record. 13 showed familial relationships to known targets. One of those familial matches came from a sample collected in Baghdad in late July. The sample, a drinking glass retrieved from a meeting room used by Quds Force operatives, showed a genetic relationship to Hossein Taib, the IRGC intelligence chief. The relationship was close, parent-child or sibling-level confidence. Yael reviewed the intelligence file on Taib. He had 3 known children, all of whom lived in Iran and rarely traveled. The Baghdad sample didn't match any of them. That meant either Taib had a 4th child Mossad had never documented, or the sample belonged to a sibling. Shamir's lab ran an age estimation analysis based on telomere length and epigenetic markers. The DNA suggested the individual was between 35 and 45 years old. If it were a sibling, it was someone close to Taib's age. If it were a child, it was someone Taib had concealed from public records. Yael made a decision. She instructed analysts to assume the Baghdad sample belonged to an unknown relative with operational significance. The profile was flagged for priority tracking. If another sample from the same individual surfaced, it would indicate travel patterns that could be used to predict future meetings. It was a reasonable assumption. It was also wrong. In early August, a second sample was collected in Beirut from a cafe frequented by Hezbollah officials. The DNA matched the Baghdad profile. The same individual had been in Baghdad in late July and Beirut 2 weeks later. Yael interpreted the pattern as confirmation. This was someone moving between IRGC and Hezbollah command structures, likely serving as a courier or intermediary. She drafted a report for the operations division, recommending surveillance on the individual if he could be visually identified. The report included the genetic profile, the travel pattern, and the familial link to Taib. It described the subject as a previously undocumented operative within Taib's immediate family network. The report was circulated internally. 3 days later, Shamir sent Yael a message requesting an urgent meeting. When she arrived at the lab, Shamir was standing at a workstation reviewing sequencing data on dual monitors. He didn't look at her when he spoke. The Baghdad sample was contaminated. Yael asked him to repeat that. Shamir pulled up the analysis. The sample had been processed as a single profile, but more sensitive sequencing revealed it contained genetic material from 2 individuals. The contamination had occurred during collection or transport, likely from the operative who retrieved the glass, or from residue left by someone who handled it before the target. The lab had separated the 2 profiles. One matched Reza Falazadeh, a Quds Force deputy whom Mossad had been tracking since 2017. The other profile, the one showing a familial relationship to Taib, was present in much smaller quantities, suggesting it came from incidental contact, not direct use of the glass. Yael asked the obvious question, which profile matched the Beirut sample? Shamir showed her the data. The Beirut sample matched Falazadeh, not the unknown relative. The individual Yael had flagged for priority tracking was Falazadeh, someone Mossad already knew. The unknown relative had never been to Beirut. The travel pattern Yael had interpreted as operationally significant didn't exist, and the report she had sent to the operations division was based on a contaminated sample that had been misread as a single profile. Yael pulled the report from circulation. She contacted the operations division to retract the surveillance recommendation, but the damage had already been registered. The genetic program had produced intelligence that appeared scientifically certain but was operationally false, and if one contamination had gone undetected, there was no way to know how many others were embedded in the database. Shamir proposed a solution: resequence every sample in the database using more sensitive methods to detect contamination. It would take 3 months, it would require additional funding, and it would mean suspending all new collections until the audit was complete. Yael brought the proposal to leadership in mid-August. The response was immediate. The program could not be paused. Iranian officials were traveling to Caracas and Moscow for meetings in September. Mossad needed genetic intelligence to confirm who was attending. The audit could happen in parallel, but the collection had to continue. Yael argued against it. If the database contained contaminated profiles, every new sample cross-referenced against that database could produce false matches. The program would generate intelligence that looked credible but couldn't be trusted. She was overruled. The decision came from the deputy director of the Intelligence Directorate. Operational requirements outweighed methodological concerns. Mossad couldn't afford to stop collecting intelligence while it verified the reliability of the intelligence it had already collected. Yael returned to her office and drafted a memo documenting her objection. She sent it to 3 people. None of them responded. The next day, she authorized collection, operations in Caracas and Moscow. Field teams were deployed with the same instructions: retrieve any item that might contain biological material from individuals associated with Iranian delegations. Within 2 weeks, 12 samples were collected. 8 were sequenced. 4 matched profiles already in the database. 2 were flagged as familiar relationships. 1 was entered as a new subject, and 1 came back as a mixture, genetic material from at least 3 individuals, impossible to separate cleanly. Yael stared at the sequencing report for 20 minutes. Then she sent a message to Shamir. "How many samples in the database do you estimate are contaminated?" Shamir's response came an hour later. "I don't estimate. I measure, and I haven't measured them all." That night, Yael drafted a second memo. This one didn't document her objection. It recommended termination of the genetic program because the data it produced could not be operationally validated without external corroboration, which defeated the purpose of using genetics as an independent intelligence source. She didn't send it. Instead, she scheduled a meeting with the deputy director for the following week. Before that meeting could happen, Iranian state media published a brief report accusing Israeli intelligence of conducting biological espionage in several Middle Eastern cities. The report provided no details. It mentioned hotels and restaurants. It described methods consistent with genetic collection. Yael read the report twice. Then she opened her secure laptop and began drafting a third memo. This one was an abort recommendation. The abort recommendation was never formally submitted. By the time Yael finished drafting it, the decision had already been made for her. On August 22nd, Hezbollah's security apparatus detained a Lebanese hotel employee in Beirut. The employee had been providing low-level information to a contact he believed worked for a European consulting firm. During interrogation, he described requests from that contact over the preceding 6 months: photograph specific guests, note their room numbers, and save any items they discarded, particularly glasses, cups, and tissues. The employee didn't understand why. He thought it was corporate intelligence work. Hezbollah understood immediately. The interrogation report was shared with Iranian intelligence within 48 hours. By early September, Iranian officials traveling abroad began using disposable items brought from Iran, sealed water bottles, prepackaged utensils, and personal towels stored in diplomatic pouches. Hotels frequented by Iranian delegations received instructions from security officers accompanying the delegations. Incinerate all waste from specific rooms, prohibit cleaning staff from handling certain items without gloves, and report any unusual interest from employees. Mossad didn't learn about the Beirut detention until mid-September, when signals intelligence intercepted a conversation between IRGC officers discussing biological security protocols for overseas travel. By then, field collection had already collapsed. Operatives in 3 cities reported that Iranian officials were no longer leaving retrievable material behind. In Istanbul, a field team observed a security officer collecting used glasses from a restaurant table and placing them in a sealed container before leaving. In Damascus, hotel cleaning staff reported being denied access to rooms until Iranian guests had personally disposed of all waste. The window for genetic collection had closed. Shamir's audit of the database concluded in October. Of the 53 profiles entered between January and August, 11 showed evidence of contamination. 4 were mixtures that had been processed as single profiles. 7 contained secondary DNA from handling or environmental exposure. The contamination didn't invalidate the profiles. It meant they couldn't be reliably cross-referenced without additional verification. Yael was removed from operational oversight of the genetic program in early November. The decision was described internally as a reassignment. She was transferred to a strategic planning role within the Intelligence Directorate, a position with no field authority and no access to active collection operations. She didn't contest it. The genetic program continued under new management, but its scope was reduced to passive acquisition, retrieving samples from cooperative sources who had legitimate access to discarded materials, such as medical waste from clinics used by Iranian officials or items left behind during inspections at border crossings. The yield was minimal. By mid-2020, the program was producing fewer than 3 usable samples per quarter. What the program had produced, the database codenamed Aleph, remained active. 19 confirmed profiles, 12 familiar relationships, 8 travel pattern correlations that had been verified through signals, intelligence. The data wasn't complete, but it was usable. Mossad had mapped a portion of Khamenei's inner circle that had been invisible before January 2019. In August 2020, Abbas Nilforushan was killed in a targeted strike in southern Beirut. The strike was never officially attributed, but intelligence sources confirmed that Israeli surveillance had tracked Nilforushan's movements in the days prior, using a genetic sample collected from a towel in a Damascus hotel 3 weeks earlier. The sample had placed Nilforushan in proximity to Hezbollah commanders planning operations against northern Israel. The genetic program had delivered a tactical success, but it had also delivered a doctrine to Iran. By late 2020, Iranian security services had begun implementing biological security measures across their foreign operations. Operatives traveling abroad were trained to treat all personal items as intelligence vectors. Hotels, restaurants, and airports in cities frequented by Iranian officials reported new waste disposal protocols that treated ordinary trash as classified material. Mossad had proven that invisible evidence could be weaponized. Iran had learned the same lesson. Yael left Mossad in February 2021. She declined to discuss her reasons, and the agency provided no comment. The Aleph database remains classified. The genetic program exists in limited form, now managed by a joint unit within the Intelligence Directorate and the IDF's Medical Intelligence Division. What Mossad discovered through the operation wasn't just a method for tracking targets. It was a truth about modern intelligence work. The most valuable evidence is often the evidence your adversary doesn't know they're leaving behind. But once your adversary understands what you're collecting, the method becomes a signal. And in espionage, signals are vulnerabilities. The operation succeeded in identifying individuals Mossad had never tracked. It clarified relationships that the agency had misunderstood. It enabled at least one strike that wouldn't have been possible otherwise. But it also taught Iran what Israel could do. And teaching your adversary your capabilities is often more costly than the intelligence you extract. The program didn't fail, but it didn't remain invisible. And for Yael, those outcomes were indistinguishable. If operational intelligence interests you, Hidden Ops covers the methods most documentaries avoid, stories built from tactical decisions, not narratives. Subscribe to see how intelligence works when success and failure occupy the same operation.